Now we're circling back to talk about this process known as osmosis. And remember that osmosis is a type of passive transport, meaning that we do not need ATP in order for it to occur. Now, one reason why osmosis is kind of different is that so far we've been talking about solutes moving across a cell membrane. But in this case, we're talking about not the solute moving, but the solvent. So we're actually going to be talking about water moving from one area to another area. Because if we take a look at our cell membrane here, water is able to cross the phospholipid bilayer of the cell membrane. So all cells are permeable to water. But remember that sometimes those solutes are stuck and they can't cross the cell membrane. And so they might be stuck either outside the cell or potentially inside the cell. It just depends on what the condition is. So in cases where the solute cannot cross the cell membrane, sometimes the water will cross the cell membrane in order to kind of balance out the concentration between the two areas. Because remember that the cells are kind of ultimately trying to get to this concept known as equilibrium, which would be equal concentrations on each side of the cell. And remember that the concentration of a solution is equal to the amount of the solute in the amount of the solvent. So if we have areas with different concentrations, we can either move the solute from one area to another area, or if we can't move the solute, we could move the solvent. So you can kind of think of the movement of the solvent as kind of like diluting the more concentrated area. So think of this as a dilution. Rather than removing solute, we are going to dilute the more concentrated area. So if we look at this example here, we can see that we have this kind of semi-permeable membrane in between these two areas. And we can see that this area on the right is a higher concentration area, and this area on the left is a lower concentration area. Now, in this example, the solute here is not able to cross that membrane. So the solute is stuck where it is. So in order to even out these concentrations, instead of the solute moving, instead the water is going to move from the low concentration area into the higher concentration area over here. So the water is actually going to move from the left side of this U-shaped tube into the right side. And in doing so, the volumes are going to change, but the concentrations are going to equalize. So the volume changes, and in doing so, the concentrations reach equilibrium. Because we don't care about the overall volume, what we care about is the difference in concentration and trying to reach equilibrium. So even though this side has a higher volume of water, and this one has a lower volume, they have the same concentration. So the concentration on this side is equal to the concentration on this side. So cells have a water homeostasis. So we can call this the water balance or the homeostasis. And remember that homeostasis is typically a good thing. This is something that our body wants to maintain. So when we look at a cell and we look at the intracellular fluid compared to the extracellular fluid, homeostasis means that the concentration of the extracellular fluid should be the same as the concentration of the intracellular fluid. Because remember that water can freely move across the phospholipid bilayer. And so if these concentrations are different, then water is either going to move into the cell or it's going to move out of the cell in bulk, which is going to change the size and the shape of the cell. So there are three possible types of conditions that can refer to the extracellular fluid, and specifically the concentration of the extracellular fluid. And so the extracellular fluid can either be isotonic, hypertonic, or hypotonic to the intracellular fluid. And we'll take a look at what happens in these different situations on the following slides. The first condition that we're going to look at when it comes to water balance in cells is this isotonic condition. And in an isotonic condition, it means that the extracellular fluid, so the fluid that is outside of a cell, is going to be the same as the concentration of the fluid inside the cell. So if we look at these little brackets here, so these brackets mean concentration. So this is just a shorthand way of writing the word concentration. And if we look at this word isotonic, so iso means same. So we're looking at the same concentration between the fluid inside the cell and the fluid outside the cell. And this is our homeostasis. So this is what our body is striving for, and this is our body functioning the way that it should. Now, there's still going to be some movement of water in and out of the cell, but we have a one-for-one exchange of essentially one molecule of water enters the cell for every molecule of water that exits the cell. So we say that there's no net movement of water because that means that we're not increasing the amount of water in the cell, or we're not decreasing the amount of water in the cell, but the amount of water in the cell is staying the same. Another condition that may exist is the hypertonic condition, and hyper means too much. So in a hypertonic condition, it means that the extracellular fluid of the cell is going to be more concentrated than the intracellular fluid of the cell. So what that means is that if there's more solute outside and there's less solute inside, and remember that the solute can't move, but the solvent can. So what's going to happen is that water is going to leave the cell. So the water is going to go towards the extracellular fluid, and it's trying to dilute that more concentrated extracellular fluid. Now, the problem with this is that the cell is going to shrivel. So we can see here that these are supposed to be nice, round red blood cells, but you can see that they're shriveling. And that's because as the water leaves the cell, it causes the cell to shrivel. And a shriveled cell can't do its job and can lead to health problems or even death. The third possibility is a hypotonic solution. And hypo means too low or not enough. So in this case, the extracellular fluid is lower in concentration than the intracellular fluid. So that means that there is too much solute inside the cell and not enough in the extracellular fluid. Now, water is going to enter the cell because remember, the solute can't move in this example, but water can. So water is going to enter the cell because it's going to try to dilute the more concentrated intracellular fluid. All right, so the water is going to move from outside the cell into the cell. Now, this is also bad. So this is not homeostasis. What happens is that the cell starts to swell up like a water balloon, and it might actually burst open. And a swollen cell or a busted open cell is not going to be able to fulfill its functions the way that it should. So let's do kind of an overview of this tonicity and what happens when the concentration of the intracellular fluid is not the same as the concentration of the extracellular fluid. So let's say we have 3 beakers. And these beakers have different solutions in them, and these solutions have different concentrations. And let's take a red blood cell and put one in each of these different solutions. So first, let's look at our isotonic condition. In our isotonic condition, remember that this is our homeostasis. So this is the one that our body is trying to maintain. And with an isotonic condition, what happens is that the concentration inside the cell is going to be the same as the concentration outside of the cell. So for example, if let's just give an arbitrary 9 grams per liter of a concentration of solutes inside the cell. That means that outside the cell in the extracellular fluid, it also needs to be 9 grams per liter. So in this situation, you have the concentration of the extracellular fluid is the same as the concentration of the intracellular fluid. You do have some water entering and leaving the cell, but it's going to be happening in a 1-for-1 exchange. So there's no net movement of water. So this is our homeostasis, which means that the cell is at equilibrium. Our next possible condition that we talked about was hypertonic. Remember that hypertonic, hyper means too much. So in this case, the extracellular fluid has too much solute in it. So if we keep the intracellular concentration the same, we can make an arbitrary extracellular concentration, and we'll just say that it's 20 grams per liter. Now remember, water is moving across the cell membrane. We've got some moving in and some moving out. But remember that the water is going to be moving towards the more concentrated area because during osmosis we are going to dilute the more concentrated area. So what happens is that we end up with more water leaving the cell than we had water coming into the cell. All right, so in this condition, the extracellular fluid is more concentrated than the intracellular fluid. Water is going to leave the cell. This causes the cell to shrivel, which is not ideal. And this is what happens when you become dehydrated. So when you become dehydrated, you're losing fluids from your body. And as you're losing those fluids, your body fluids are becoming more concentrated because you're removing water from these solutions. So when you become dehydrated, your extracellular fluids might start to become hypertonic to your intracellular fluids, which means that water is going to start leaving your cells to dilute the hypertonic solutions, and your cells are going to shrivel, and shriveled cells aren't going to be able to do what they need to do. And so if you get too dehydrated, it can actually lead to death because your cells are not able to function. The next condition we're going to talk about is our hypotonic. Remember, hypo means not enough or too low. And so in this example, we're going to keep the intracellular concentration the same, but let's say that this the cell is now put in pure distilled water with a concentration of 0. So remember that we've got water that's going in and out of the cell. But now the more concentrated area is inside the cell. So what we end up with is water moving into the cell in order to try and dilute the more concentrated area. So in hypotonic, the extracellular fluid is less concentrated than the intracellular fluid. Water is going to enter the cell. The cell is going to swell or potentially burst like a water balloon. And this is obviously not good. So a swollen or bursted cell is not going to be able to carry out its function. Now, the good news is that this is fairly rare, and that's thanks to your kidneys. So as long as your kidneys are working properly, it's pretty hard to drink too much water. And that's because as you're drinking water throughout the day, your kidneys are just going to get rid of the excess water. Now, there is a possibility of something called water toxicity. And this can happen if you drink significant amounts of water in a short period of time. It can also happen in athletes because in athletes, they're losing both water but also solutes. And if they're not replenishing the solutes and just drinking pure water, then it can actually lead to this hypotonic condition, which can be dangerous and potentially fatal. So for the average person drinking water throughout the day, you are probably not going to encounter a hypotonic situation. But there are examples of people that have had too much water to drink, and if you essentially dilute your extracellular fluids too much, it can lead to serious complications and potentially even death. But most likely, you are much more likely to have a problem with dehydration. So go ahead and drink that water.